Great. So hello everyone, and many thanks for joining this live speaker Q&A. It's my pleasure to welcome the speakers to this session. Attendees, please do comment any questions on the platform via the chat feature, and our speakers will be happy to answer them. You're also welcome to join the Zoom call and ask your questions to the speakers directly as well. So let's get started. Our first question is for Emily Ann. Which is, what are the steps you take for validation of multicolour flow cytometry assays across flow hubs or sites?
So thankfully, we do a lot of our flow cytometry analysis at one site, so we get to avoid a lot of the cross-site validation. But we have had some instances where we need to run our assays at another AstraZeneca site. And in that case, if we know that we need to do that, we usually do some work in terms of checking that the voltages are similar, and we do a kind of preliminary setup study just to make sure that everything translates really well, and where we're expecting to see populations, we do see them. But luckily that would just be analysis as well. It wouldn't be the actual setup and staining of the cells. So we do save a little bit of time and setup in that case.
Great, thank you so much for that. Stephanie, would you be able to describe to us some of your priorities over the next year?
Priorities in terms of?
In terms of your research in this area, what are your next steps?
So a large area of growth for us is looking at immunometabolism, because it allows us to combine two of the big core strengths of the technologies we have. So using things like the mass spectrometry imaging to look at the spatial resolution of endogenous metabolites, and using the imaging mass cytometry to look at all of the different immune populations and their distributions, and combining those two. And I say immunometabolism, I mean sort of applications of that in a wide variety of different therapeutic areas. So we support oncologies that would be looking at IO. We support our respiratory and immune partners that would obviously be looking at the immune there, but also looking at some of our cardiovascular and metabolic diseases and the inflammation events that happen in those, and how that cross-talks with metabolism.
Fantastic, thank you for that. It looks like Lorna is having a bit of technical difficulty, so hopefully she'll join us soon again. But for now, back to you, Emily Ann. I have a question for you, which is, do you think in vitro systems will become more widespread in translational research?
I really hope so. I think that the model that we've developed for the bone marrow has been really useful in terms of bridging that gap in translation from the rat to the clinic. We're hoping that we're always focusing on the 3 R's, reducing, and in terms of our animal studies. So we're hoping that this would reduce the need for some animal studies if we could get this bone marrow model really well translated. So yeah, I really do hope that at least our bone marrow model, it would be used quite often in the future.
That's great. Back to you, Stephanie. What are the advantages of multi-parameter flow cytometry tools instead of singleplex?
So for us, it's really that you get, as particularly around systems like the immune system, rather than looking at each individual marker by themselves, you're looking at the interaction of multiple different cell types. You can, as well as identifying the types, get their activation state, their polarization states, any slight signaling changes. You really get a much bigger wealth of information. And when we're looking at that with the spatial context, you also build in how they're interacting, what compartments they're in, and how this relates to all of our different other modality phenotypes as well. So you get much richer, deeper, more mechanistic insight from that type of analysis.
Absolutely, thank you for that. A question for both of you, but we'll stay in order as we have been. What do you think are the biggest challenges yet to be solved in flow cytometry? So Emily Ann first.
I think for us, I know it was mentioned in an earlier talk, just how many parameters we're having to deal with in terms of data analysis. I know for us, we're kind of topping out in terms of gating hierarchies. It really, we need to have some way of that dimensionality reduction in the data analysis. So I think that's the biggest challenge that we're facing, and probably a lot of other teams are facing too.
Fantastic, thank you. And Stephanie, the biggest challenges that you're considering at this stage?
I would agree, to be honest. It's around that analysis part. So even in the imaging space, we segment and treat the cells as single cells, but we did some calculations looking at the number of possible phenotypes when you are looking at 40-plex data sets, and it becomes, I think, more than the number of stars there are in the universe by a factor of the number of stars in the universe. So it's a ginormous number of potential phenotypes that exist in that data, and obviously not all of them do exist, of all of those potential combinations, and so it's developing those tools to pull out what different phenotypes actually exist within your data and how they're changing. That's is a big focus for us, and I think that's the biggest challenge, to do that accurately and robustly.
That's great. Thank you so much, Stephanie. Lorna, I can see, has joined us again. We do have a few questions for you, Lorna. The first
I'm so sorry.
No, that's okay. No
I'm here
problem.
now.
I'm glad you can join us. Yeah. The first question is, when optimizing stimulation conditions, what other stimulants were tried before settling on T-cell Transact as the best stimulant?
Yeah, so it's a really good question. And actually, for the work that we were doing, we were basing it on some pre-work had already been done by some of the research groups, and so we went with Transact. So in my hands, I didn't actually test any others in the situation that I was showing you. But in other work, we've also looked at using Dyna beads, so you can use beads to stimulate T cells. So that's one option. The can be a little bit more technically challenging because you're then introducing a bead population into your cell population, but you do get really good stimulation of the T cells with Dyna beads. So that's something to think about. And then another thing that we've looked at is just using, so Transact is a pre-made mix, but they have anti-CD3, anti-CD28 in there. So we've actually just been looking at antibodies which are anti-CD3, anti-CD28, and just adding those directly to the cells, which can also be a good stimulant. So yeah, and we do see it depends on the actual what you're actually looking at. The stimulant might depend which gives you the better result.
Fantastic, thank you, Lorna. And another one straight away for you. Could you talk us through what are the next steps for your research? What are your goals for the next year or so?
Yeah, definitely. So, so my talk, I mainly focused around the pooled screening, so pooled CRISPR screening, and how we used Flow for that. But I did mention that we have this automation platform, and we're trying to use that to do arrayed screening. So that's plate-based screening also with a flow cytometry endpoint. So really embedding that into our workflow is the goal for over the next year. So that platform will be arriving this year, and I think I mentioned the nice thing about that is that in the same lab we have our high throughput flow cytometers, so really marrying up that automated cell preparation and automating the preparation for flow, so the wash and stain steps, and then linking that in with our high throughput flow cytometers, so having a real high throughput flow capability which can support both the CRISPR workflows, which I was talking about, but also drug discovery workflows. So that's kind of our goal for the next year.
Fantastic, that's great. Thank you, Lorna.
No
so we've got a few minutes left of this Q&A. Stephanie, you mentioned earlier talking about the potential of flow cytometry, looking at the promise. I'd love to ask you all what you're most excited about for the future of flow cytometry. So Stephanie, should we start with you?
So I guess for me, the potential is how we combine these with different analyses. So at least within the imaging space, the imaging mass cytometry doesn't give us all of the information within that system with just one modality. So combining that with different types of modality and bringing together all of the disparate types of information you can get from these types of single cell analyses. So when we're looking at single cell transcriptomics, proteomics, signaling, epigenetics, all of these factor into how the system is behaving, and being able to bring all of that disparate information together. I think we've now getting to the stage where we have the tools to be able to do that type of analysis, and I think that's very exciting.
Absolutely. And Emily Ann, what are you most excited about at the moment for flow cytometry?
I think I'm going to echo some of what Stephanie said, bringing together several different modalities. Within our team, we have done some single cell RNA-seq characterization of the bone marrow model, and we're also looking at some Cytoph as well. So I think, you know, having all of these different abilities and then bringing them together to better define a good flow cytometry panel would be great. And we're also getting a spectral flow cytometer this year, so that's my current exciting development. So I'm looking forward to building a panel around those previous new analyses we've been doing.
Fantastic, absolutely. And Lorna, would you agree with what's been said so far? Do you have any other areas that you're excited about at the moment?
No, I think you've covered that really nicely. I think those are the really exciting areas. And I guess, as I said, one of the things that we're aiming to do is to try and make this more high throughput flow cytometry accessible to multiple different projects within GSK. So I guess that also is something that's exciting, trying to make it more amenable to more different screening applications and kind of get more people using it for their the question that they're asking.
Absolutely, that's fantastic. Well, thank you everyone. We'll conclude the session there. So thank you everyone for joining this Q&A, and thank you to our speakers for sharing their time and expertise as well. If you did have any more questions for our speakers, please do feel free to reach out to them directly via the platform. And thank you again. We hope you enjoy the rest of the event.
Thanks very much.
Thank you. Bye.
Bye.